Cost-effective method for microbial source tracking using specific human and animal viruses. The study describes a cost-effective method for the identification of the source of fecal urine contamination or contamination by nitrates in water using quantitative PCR for the specific quantification of human, porcine, bovine DNA viruses, adenoviruses, and polyomaviruses proposed as microbial source tracking tools. We have developed a protocol for the concentration of viruses from 10-liter water samples by organic flocculation using skimmed milk, extraction of viral nucleic acids from the sediment, and quantification of human, bovine, or porcine DNA viruses using quantitative PCR.
It is well known that microbial contamination of the environment represents a significant health risk, and we need to know the sources of the contamination. We have proposed the use of highly prevalent DNA viruses as microbial source tracking tools. The selected viruses are adenoviruses and polyomaviruses. Those viruses are being excreted persistently all over the year and in all geographical areas studied. In previous studies, we have developed robust, low-cost concentration methods for viruses in water, and we have been working developing qPCR assays for the quantification of porcine adenoviruses and bovine polyomaviruses. In addition, we use human adenoviruses and JC polyomaviruses tested by qPCR also as markers of human contamination in water.
Collection of water samples.